All right. So like we went through in the problem set, you understood what the locus meant? That's a position on the chromosome, so that's the gene. Right?
Yeah.
And that the seedlings, you know what seedlings are? They're plants that are growing out.
Yeah, like the sprouts.
They sprouts, yeah, that's exactly what they are. So now, with plants, you know that they need the chlorophyll to do photosynthesis, right? So that they can turn sunlight into food, right? Because that's how they get their food. Is that right?
Yeah.
Okay. So you understand codominance, right?
Mm-hmm.
So that CGG gives you green chlorophyll because it works, the chlorophyll works really well, and they're able to do photosynthesis well. These don't do it as well because they have the CG. So this produces the chlorophyll, this one doesn't produce the chlorophyll. It's the same gene, just different alleles. So it turns out we can see this because this is green-yellow, because this is codominance, right?
Okay.
This one can't make chlorophyll very well at all, so we only see yellow.
Okay.
To our eyes, chlorophyll is green. That's why plants are green, because they have chlorophyll. Does that make sense so far?
Yeah.
All right. So here are the observed numbers. These are what we see. When they did the cross, when they grew them, after 7 days, they had 49 greens, 111 green yellows, and 56 yellows, totaling a total of 216. These are individual plants. Each plant has 2 genes, one from its mother, one from its father, correct?
Yeah.
Just like us. So if we ask how many alleles there are in the gene pool, remember what a gene pool is. A gene pool is all the genes in a population. All right? So if these are all the plants, there's 216 of them. How many genes do you have in those 216?
The double.
Double it, right? Does that make sense so far?
Yeah.
All right. Is this helping?
Yeah, it is.
Okay. So here we know we have 216 total creatures, but the total number of genes, because they're diploid, some animals are haploid, they only have one. But these are diploid, they have 2. So then here there's 2 for each 49, so that's a total of 98. Right? 98 C big G, C big G, yeah?
Yeah.
So we go to write down how many CGs we have 98 from all the green plants. Now, this is where I made my mistake. When in class, I said 111, half of them are CG, so it's 50 something. That's what I said, and I wrote it down. This is the same mistake I made last week when I went over a similar problem with the AP kids. It's not half. There's 111. It's that we're not doubling it. There's 111 individuals with 1 CG and 1 CY. So that means there's 111 CGs and 111 CYs.
Oh.
Does that make sense?
Yeah.
This is where I made the mistake here. And that's why the numbers didn't come out the way they should have when we were doing it in class. All right, so when we add them up, the total number of CGs from, obviously this is 256 for this is CY, would be 256 times 2 because there's 2 copies of each Y, right, is 112. And there's 111 CYs here. Again, I made a mistake. I put 50-something, right?
Yeah.
It's actually 111. I felt like an idiot when I went back and looked at this. And as I said in class, these are easy to make mistakes, and you got to keep everything straight, okay?
No.
So 112, so you have a total CGs, of all the CGs in all 3 of these populations, 209. The CY total is that 112 plus 111. There's no CYs in the green, right?
Mm-hmm.
So altogether you have 223 alleles, CY alleles. You have a total of 209 CG alleles. So 209 divided by 432. Why 432? Because there's 216 individuals with a total of double that for the number of alleles. So your P, which is the probability of, or how many, with the frequency of the CG is 0.4840. That's these two this one divided by that one.
Okay.
The probability or frequency of CY is 223 divided by 432, which is 0.516. That's Q. These two together have to equal 1. Remember in class I tried to add them together, they didn't equal 1. I was like, we'll come back to this later. I didn't want to waste time in class, and this is where I made my mistake here. I have these, I should not have. It's just I don't double it, right? Here I doubled it because there's 2 copies of each, but here I'm not going to double it because only one of them is CG, the other is CY.
Oh, okay.
So these two added together should equal 1. Well, add them together, they equal 1. So that's right. All right, so that gives us our P and our Q. Then we use this formula. This is the predicted. This is what it predicts. If we have that many alleles, this is the frequency of the alleles, right? Then we go ahead and put them into this equation. Again, we have a probability of 0.234 is PQ, a probability of 0.266 is Q squared, and 2PQ is the probability of having the heterozygous. So this is a homozygous big G, C big G. This is a homozygous C big Y, and this is C big Y, C big G. So these are the 3 probabilities or frequencies that we'd expect. That's how often we'd expect. They equal about 1, right? Oops. So how many do we expect? If we use this, how many would we expect? Well, P squared is how many CGG we'd expect. We'd expect about 51. This is how many. This is messed up. I messed up my notation. This should be CY. This is Q squared should be about 57. And then the GCY should be about 108. That's P plus times Q times 2, right? That's 108. All right, I'm sorry. The frequency of P times 216 should be about 108. Hold on, let me see. So it should have been 108, and what I have here is 0.499, which is 2 times P times Q. So that's not P and Q, but P and Q was from the original discussion, right? What we did a minute ago. I don't know where it went. There it is. Remember, we calculated 0.4840, 0.516 for P and Q. Then we plugged it into the equation, and we got 0.499. And then we used that times the actual number, 216, to get what we expect. This is the expected, right? What we expect to see for the outcome for the heterozygous should be 108. It should be 108. Oh, look, I did it right there. I don't know why I had 116 over there. So 108 heterozygous. We expect 57 CY, so 57 yellows and 51 greens. After 7 days, this is what we expect. This is what we observed. So they're pretty close, yes?
Yeah.
And is this difference significant? Well, we'd have to do a chi-square because it's so close. It's hard to suspect if it's significant or not.
Oh, okay.
But if you think about how plants work, you'd expect that this has a disadvantage because it's not green. These are yellow. They don't have chlorophyll. So you'd expect that they wouldn't last very long. But seeds bring them with them food, right? In seeds, the mother plant puts food so that the seedling can grow initially before it can start to do its own photosynthesis. But you'd expect that it would grow some, but you'd expect that they would have a weakness. But it looks like they're pretty close to what they were after 7 days. Well, it turns out their food starts to run out after like 20 days.
Oh.
So now we start to look at what we observe after 20 days. After 20 days, After 21 days, this is what we see. This is how many there are of the expected. So we'd have to look and see how many we expect to see if there was only 173. Because if you look at the problem, it says there's 100, now there's 173. With these many, 47, 106, and 20 is what they observe out of those 173. We do the same thing we did a minute ago, but this, we don't have to do all that, all those predicted frequencies, because we know what the frequency should be. There they are. This is what we already calculated, right?
Yeah.
That doesn't change. So the now times 173, we expect to see 40, 46, and 86. If nothing's changed, if there's no evolution going on, if nothing's being selected for, if there's nothing that says this one's better fit to survive than the other one, then we expect that this would be the percentages. The lights go off automatically, by the way. There's nothing I can do about it. Now, this is what we expect to see. This is what we got. Now, remember what we just said. The green plant, we think, should have an advantage because it's green. It can do photosynthesis. This can do a little photosynthesis. That's why it's kind of green-yellow.
Yeah.
This one's yellow. It can't do photosynthesis. So after it eats up all its food that it had in the seed, we expect that they should start dying. Well, this is what we expect to see. There's a lot less.
Yeah.
And there's a lot less than we would expect to see if it was no evolution, if there was no advantage, right? Because a large population, no migration, no mutation, random mating, and no advantage to either any of the alleles, then we'd expect to see this. But this is what we get. So there was evolution, and we know that this one has an advantage because we just discussed that photosynthesis allows it to make its own food. So these two are a lot less. So evolution is occurring, and the yellow and yellow-green are being selected against. So they're dying.
Yeah.
And the green ones are surviving. About what you'd expect, right? In fact, a little more, a little better than if no evolution is going on. So what we say is that CY individuals cannot produce chlorophyll, so they can't do photosynthesis. In the first 7 days, the seedlings live off the stored resources in the seed. By week 21, the resources are depleted, and CY, the yellow plants, are dying.
Yeah.
And that explains why we see what we see. So that's just a real-life look at how you can use this formula to predict an outcome. Now, I'm not going to ask you to do this on a test, okay? So don't worry about it. But I think it's important that you understand that it is possible to do an analysis using Hardy-Weinberg of a population and decide, does one allele have an advantage? And as a doctor someday, right, you might look at a population and say, wow, we found this gene in this population. Is this gene an advantage or not? And so you can count how many there are, how many would if there was no evolution going on, how many you'd expect to see, and then ask yourself, is that what we got, or do we have more? If you have more than what you expect, then maybe there's an advantage to the gene. If you have less than what you expect, maybe there's a disadvantage to the gene. And then that could lead your research in that direction. You could start figuring out why that gene is more fit to better, gives that individual an advantage. Does that make sense?
Yeah.
All right. So that's all there was to that.
Okay, I understand it now.
All right, very good. Thanks for coming by.
Yeah, no, thank you too.